What's up, everybody? This is my second installation in what's becoming my new series of videos on biology, chemistry, and my other various science classes that I'll be taking. Today's video is going to be about DNA and its structure and why it is structured in such a way and why it does the double helix thing and bends. First thing you need to know about DNA is that there are three main components in DNA. There is phosphate, sugar, and then a nitrogenous base. Now, they come together to form a nucleotide, which will look like this. This is the phosphate right here. The black part is the sugar, and then this is the nitrogenous base. And these, what happens is they get a bunch of these and they all hook up together into two big chains that connect down the middle to make this what looks like a ladder. And then of course the way the bonds come together, that's what makes it form the double helix, just like that. So if you look at this, you've got your phosphates and your sugars going all the way down, because they make up the backbone of what DNA is. And then if you look in the middle, you've got these rungs of nitrogenous bases. There are 4 different nitrogenous bases. They are abbreviated by letters A, C, G, and T. So the actual names for these nitrogenous bases are adenine, cytosine, guanine, and thymine. And the bases are basically split into 2 pairs. Adenine can only be bonded with thymine, and cytosine can only be bonded with guanine in DNA. And that's where you get these cool little color-coded things. Adenine with thymine and cytosine with guanine. And so they they bond together just like that. Now, if you'll notice by looking at my little model, there's an extra phosphate on this end on the bottom, and then an extra phosphate on this end at the top. And that's because these are actually facing 2 totally different directions, and because that's how they come together. Because when they split to replicate DNA, they just split right down the middle like this. The hydrogen bonds are broken because, just like a zipper, they come undone, and then a new side exactly like this side is formed to go with this side. So as you saw when I split this little bit of DNA in half, there were these little white things sticking out of the nitrogenous bases. Those are actually meant to represent the hydrogen bonds that are formed between the nitrogenous bases. Now, I've got another model that is better to see this. Here we go. This is also DNA. You got your phosphates, and then your sugars, your bases, and then these represent the hydrogen bonds. Now, as you can see, one of them has 2 hydrogen bonds, one of them has 3 hydrogen bonds. That would be cytosine and guanine have the 3 hydrogen bond, and then adenine and thymine have the 2 hydrogen bond. So the reason that there are 2 hydrogen bonds between adenine and thymine and 3 between cytosine and guanine are simply because of the chemical structure of each of those base pairs. The way it lines up, it makes it so that only cytosine can bond with guanine because it's the only base pair that shares 3 possible hydrogen bonds, and so they bond together at the 3, and then adenine and thymine bond at the 2. So now that I've informed you of all these things, it's all those little bonds and reasons that DNA sets up the way it does, that it works the way it does, and why it will eventually end up looking like this, our most complicated model. As you see, it's got the whole double helix going on, and it's got one big ridge right here, well, better up here, but right here it's got one big ridge and then one smaller ridge, because this is where proteins come in and latch on to do their thing with DNA and various other things. It's just all these little things about DNA and its structure that make it what it is and why it works for us.